Welcome to today's presentation. I'm excited to be introducing our guest speaker in his presentation titled Circulating Tumor DNA for Minimal Residual Disease Research by Hybridization Capture for Resected Cancer Samples. In this webinar, we'll explain how IDT's HiCapture solutions are assisting researchers with overcoming challenges associated with circulating tumor DNA for MRD research. You'll also learn more about what MRD is, how it compares to traditional methods, and the impact it is making with discovery of new biomarkers. We hope by bringing these studies to the spotlight, it will foster new collaborations and discoveries within oncology research. Now, in regards to our speaker, Jerry received his bachelor's in biotechnology from the University of Toronto and obtained his master's from Brock University in 2021. He currently works at GeneSeq Technology as a bioinformatician, tackling all sorts of oncology-related problems. His current areas of interest are in genetic markers for drug resistance, MRD detection, and early detection of cancers. As for myself, I'm Jordan Young. I'm the Strategic Marketing Manager here at IDT, and I lead our Collab Network program, which allows us to bring to the spotlight the work of our customers and researchers around the world. Now, without further ado, I'll pass today's presentation over to Jerry. Thank you.
Now, before I begin, I do need to give a bit of a disclaimer on some of the research that you'll be presented with today, specifically that the findings were not used to guide or to aid any diagnosis, treatment, monitoring, or screening of any disease. So for today's presentation, I'm going to give a bit of background on MRD, followed by our research highlights, and then finally I'll give a quick summary. So just a bit of background before we really get into our research. What traditional cancer care narrows down into is basically surgery through resection, followed by adjuvant therapies such as chemotherapy, radiotherapy, or I guess more recently immunotherapy. And that's all very well and good, but the efficacy of this kind of cancer care is not guaranteed in all patients. And what our research is trying to achieve is to, by looking into these non-invasive approaches such as minimal residual disease, we're trying to assist these cancer care methods in order to improve their efficacy. And we've shown some promising results. So I've mentioned MRD a lot in these past slides. What exactly is MRD? MRD, again, stands for minimal residual disease, and it basically refers to the minute amount of tumor cells that remain after the standard cancer treatment has ended. And these remaining tumor cells can divide and grow as normal tumor cells are known to do, and they release their tumor DNA into the bloodstream as well as cause resurgence of the disease. Now, traditionally, the recurrence of the disease is confirmed through radiological imaging, i.e. CT scans. And kind of the main research goal that we're trying to do is we're trying to understand how MRD analysis can compare to these traditional methods when it comes to predicting disease recurrence. To put what I've been talking about into a more graphical format, I've adapted this figure from a paper published in 2020, which compares between before MRD analysis and after MRD analysis. So over here on the left side, we can see that after treatment has ended, there still remains a small amount of cancer cells inside the body, which is essentially invisible to imaging detection. However, as those cancer cells grow and divide, they then reach a threshold at which point they can then be detected by CT scans. And what we're kind of trying to achieve with MRD analysis is we're trying to lower that threshold of detection to a point in which we can then detect that trend or that potential of recurrence earlier than what normal imaging detection will allow us to do, at which point it would give the physician more time to develop personalized treatments for the individual. And what enables us to do this type of research is a concept known as cell-free DNA, or cfDNA, which I'm sure most of you have either heard of or worked very closely with. And essentially what that is genetic material that enters the bloodstream through a variety of ways, such as apoptosis or necrosis. And the special thing about this is that we can obtain these samples through a very non-invasive method of blood sampling, which allows us then to obtain samples very frequently and at a lower risk to the patient. Now, within this giant scope of cell-free DNA, we have a smaller area called the circulating tumor DNA, or the ctDNA. And essentially what that is a specific type of cell-free DNA that originates from tumor cells. And these ctDNA have been known to carry the unique variants that we find within those tumor cells. And a variety of papers have already been published on the utility of ctDNA in multiple cancer cells, such as breast cancer, colorectal cancer, gastric cancer, etc. But today we're mostly going to be focusing on lung cancer and colorectal cancer. So a quick little side note on the methods that we'll be using. We'll be focusing mostly on targeted panel sequencing, with the first one being the ATG-seq 139 gene panel, which covers roughly 130 kilobase pairs of the genome. Now this panel uses a bi-barcoding system, as well as can sequence up to around 30,000 times coverage for a plasma sample. On the other hand, we have the GeneSeq Prime 425 gene panel, which can sequence roughly 5,000 times coverage for a plasma sample. And of course, our research here wouldn't be possible without our collaborators over at IDT, who assisted us with our probe design, as well as their X-Gen line of products. Now let's talk about some of our research highlights that we obtained over the past year. So for our first study, we decided to take a look at lung cancer, which we all know as the leading cause of cancer-related deaths worldwide. Now, approximately 85% of that has been attributed to specifically non-small cell lung cancer. And the goal of our research here was to kind of evaluate the use of targeted next-generation sequencing on ctDNA in order to track and analyze that MRD in samples collected after cancer care. For our methods used, we obtained probe designs from IDT, as well as using the ATG-seq panel for targeted sequencing. We designed this hybrid capture panel in order to target variants in matching tumor samples that were previously obtained from a separate study, and in total 412 plasma samples were collected for the study. Now, before I move on with the results of our study, we need to take a moment and define what we mean by ctDNA positive and ctDNA negative, as that's actually subject to change between studies. Now, in this case, we define plasma samples with at least one true variant or one true tracked variant as ctDNA positive. And what we observe in this retrospective analysis of response was that, as can be seen in Figure A and Figure B, the survival curves, we see that post-surgical ctDNA status and post-adjuvant chemotherapy ctDNA status, the blue line, which represents the ctDNA negative group, actually has a higher recurrence-free survival than that of the ctDNA positive group, which is the red line. Essentially, what that means is that at any point after surgery, the proportion, I guess, of subjects or samples that have experienced relapse is lower for the ctDNA negative group than for the ctDNA positive group. And that's true for both post-surgical and post-ACT groups. Then we decided to take a look at whether the adjuvant chemotherapy status can affect the results or the rankings of the groupings. And what we found was that regardless of the ACT status, the ctDNA negative groups always comes out above the ctDNA positive groups. And then finally, we also looked at kind of the serial surveillance of longitudinal ctDNA. And what we found was that there's a significant difference between the recurrence-free survivals of the ctDNA negative group versus the ctDNA positive group. Following that, we also took a look at CT detected recurrence versus ctDNA detected recurrence. And what we found was that for MRD analysis via targeted sequencing, we were able to identify tumor-associated variants on average 88 days before CT scans could detect the recurrence. So to quickly summarize study 1, we found that targeted NGS approaches help with the identification of cancer-associated variants in ctDNA samples collected after cancer care. And then going back to that previous slide, we actually found that 24 research samples were identified as high risk via MRD analysis and were actually later linked to relapse via CT scans. Now moving on to study 2, which is very similar in theory to study 1, we took a look at colorectal cancer specifically, which is the third most common cancer type worldwide, with a 5-year mortality rate of 40%. Now, in terms of methods, similar to this first study, we used probe design by IDT, but this time we sequenced it using a 425 gene panel of GeneSeq Prime, and we designed the hybrid capture panels to target variants that were identified in matching tumor samples previously collected during cancer care. And in total, 720 plasma samples were collected for the study. For this study, what we defined as ctDNA positive samples were the plasma samples that had no less than 5% of their total tracked variants being defined as true variants, which were also found in the matching tumor samples. Now, from the survival analysis, we can see that regardless of the time point, whether it be post-operative, post-adjuvant chemotherapy, or serial post-definitive treatment, the recurrence-free survival of the ctDNA negative group was always higher than the ctDNA positive group. Now, following that survival analysis, what we did was compare between our ctDNA detected tumor variant versus the CT scans detected recurrence. And what we found was that MRD analysis via this targeted sequencing method was able to detect tumor variants on average 5 months before CT scans. And so to summarize study 2, in this retrospective analysis of our samples, we used MRD analysis to show that cancer-associated ctDNA variants that were detected after cancer care are statistically correlated with disease relapse. Moving on from that, we decided to take a look at some other applications of MRD within this cancer area, beyond that of just detecting cancer recurrence. And what we settled on was trying to evaluate the effectiveness of tracking variants in ctDNA in order to assess potential response to neoadjuvant chemoradiotherapy in colorectal cancer samples. Now, in terms of our methods here, we again obtained probes designed from IDT, and then we sequenced using a 425 gene panel. And then we decided to design a hybrid capture panel in order to target cancer-associated variants in plasma samples this time, as opposed to tumor samples. And in total, we procured 531 plasma samples across 5 time points, as listed in the table here. From 10.1, we have before the neoadjuvant chemoradiotherapy, which serves as a baseline for each subject, followed by after the 15th round of NCRT, and then 25th round of NCRT, and then before surgery, and then finally at time point 5, which was after surgery. To start with, we divided the groups based on response versus non-response to the neoadjuvant chemoradiotherapy. And what we found was that in genes TP53 and APC, there was a high proportion of mutations in the non-response group versus the response group. On the other hand, in pathways HRR and HMT, there was a significantly higher proportion of mutations in the response group versus the non-response group. Furthermore, after separating the groups based on their tumor regression grade, which was determined using MRI, we saw that there was a distinctive, significant upwards trend, or an increasing trend in proportion of mutations in the TP53 and the APC gene as tumor regression grade increases. And for the pathways, HRR and HMT, there was a decreasing trend in mutation proportion as TRG increased. Next, we took a look at whether the clearance of those baseline mutations in time point 2, time point 3, and time point 4 of the response versus non-response groups could be any different. And we saw that there was a significant difference in the proportion of mutations that were cleared, with the higher proportion being in the response group and the lower proportion in the non-response group. However, we saw that the non-response group did actually acquire more mutations than the response group. And then finally in figure D, we see the similar trend in which as TRG increases, there was a lower proportion of mutations that were cleared and a increasing proportion of mutations that were acquired. So moving back to our main topic, which was that of MRD analysis, we compared between the recurrence-free survival of samples at time 4 and time 5, which was before surgery and after surgery. What we found at time 4 was that the ctDNA undetectable group, which was corresponding to the ctDNA negative group, had a much higher recurrence-free survival than the ctDNA positive group. Then upon adding a predetermined list of 6 high-risk features, at time 5, we found that ctDNA positive group with high-risk feature positivity meant a much lower recurrence-free survival than that of the other groups. In order to summarize study 3, we found that our analysis of ctDNA suggests that it might be a useful tool for assessing potential response to NCRT as well as for tracking MRD. And that our analysis of ctDNA variants suggests that mutations might be useful when assessing that risk of recurrence. Now, to note here, not only for this study 3, but for the previous studies as well, we do require larger scale research studies in order to validate our findings. Now I'll give a summary of our findings. So the major take-home message of this presentation is that research into MRD will play an important role in determining its effectiveness for identifying disease recurrence, and that research studies using ctDNA have shown that it's a feasible biomarker for monitoring that residual disease. Now, as we speak, there have been many other researches into MRD analysis, including the use of epigenetics such as methylation, as well as other cfDNA features such as fragmentomics. Finally, I'd like to take this opportunity to thank IDT for inviting me to speak here on behalf of my colleagues. I'd like to especially thank the collaborators who worked with us on the studies mentioned in today's presentations, as well as my co-workers and supervisor at GeneSeq. And of course, thank you all for attending this session and for your attention.